Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everyone. My name is Abdulrazaq Albilali. I am a neurologist and headache specialist at King Saud University Medical City in Riyadh. I'm also the head of the newly established Saudi Headache chapter of the Saudi Neurology Society. And today I'm going to be talking about the pathophysiology of migraine. Here are my disclosures. Migraine is considered now as a complex brain disorder and not just a headache. This view of migraine has evolved over thousands of years ago, from first being called a sick head attack as in ancient Egypt to something caused by the unbalance of the four body humors, as mentioned in ancient Greek and Islamic literature, to something that thought to be mainly caused by the cranial vasculature system, and then finally as disorder of neuronal origin or complex brain disorder, reflecting our advanced understanding of the non-pain symptoms as well as the disability and comorbidities of migraine. Therefore, it's not surprising to know that the pathophysiology of migraine is also complex, and there is no single scheme or model that would explain all the aspects of migraine. Nevertheless, we'll try to focus in this presentation on the functional neuroanatomy of migraine and also explain the proposed pathophysiology behind each phases of migraine. So let's start by reviewing the peripheral innervation of the face and the head. So as you all know, the anterior portion of the head as well as the face are innervated by the trigeminal nerve, and there are three branches of the trigeminal nerve, often referred to V1, V2, and V3, or as the ophthalmic division, the maxillary division, and the mandibular division. Now, you can see on the right of the slide that these three divisions of the trigeminal nerve feed back to the trigeminal ganglion, and then back to the sensory nuclei of the trigeminal nerve of the brainstem. Most importantly, spinal nucleus, which has the trigeminal nucleus caudalis, which is responsible for the transmission of the pain and temperature from the ipsilateral face. Now, back of the head is innervated by the greater and lesser occipital nerves. Both of them are branches of the C2 nerve root. The medial portion of the head is innervated by the greater occipital nerve, whereas the more lateral portion of the back of the head is innervated by the lesser occipital nerve. Now, this is the trigeminal vascular system, which is considered the anatomical and physiological substrate of migraine. And as you see in this diagram, you can see the ascending pathways and the key structures in migraine pathophysiology. So starting peripherally from the sensory efferents, from the branches of the trigeminal nerve, you can see V1 and V2 and V3 project through the trigeminal ganglion and synapse at the trigeminal cervical complex, which consists of the trigeminal nucleus caudalis and the C1 and C2 contributions of the upper spinal dorsal horn. Moving to the second order neurons from the trigeminal cervical complex, which ascends in the quantothalamic or trigeminothalamic tract and synapse on the third order thalamocortical neurons. In this pathway, there are direct and indirect ascending projections also exist to the locus cereolis, periaqueductal gray, and hypothalamus. And finally, the third order thalamocortical neurons in turn synapse on a diffuse network of cortical regions, including the primary and secondary motor, M1, M2, somatosensory, S1, S2, and the visual cortices, V1 and V2. Finally, talk about a reflex connection from the trigeminal cervical complex to the superior salivatory nucleus, which projects via the sphenopalatine ganglion, and this type of projection provide parasympathetic innervation to the extra and intracranial structures, which preclinical data has shown that the activation of this projection lead to vasodilatation because of the release of neuropeptide like pituitary adenylate cyclase activating peptide and vasoactive intestinal polypeptide. In addition, preclinical data have shown that activation of the trigeminal vascular system results in antidromic release of vasoactive signaling molecules such as calcitonin gene-related peptide, CGRP. These molecules in turn also contribute to dilatation of intracranial arteries. Now, in addition to the ascending pathways, there are also descending pain modulatory pathways, which mostly responsible for pain inhibition. And as you see in this figure, there are different areas within the brainstem and the cortices that involve in this modulation. So modulation of the trigeminal cervical complex involves dopaminergic pathways through the A11 nucleus in the hypothalamus, and it is mediated by the locus cereolis, periaqueductal gray, and also through the rostral ventromedial medulla, resulting in an inhibition of the trigeminal cervical complex neuronal firing. There is also the superior salivatory nucleus, which has bidirectional connection with the hypothalamus and the trigeminal cervical complex, and stimulation of the superior salivatory nucleus lead to stimulation of the trigeminal cervical complex and increased neuronal firing within this complex. And finally, you can see that cortical spreading depression can also increase the trigeminal cervical complex neuronal activity, and this can happen directly through direct influence into the trigeminal cervical complex, or indirectly through the previously mentioned areas. So after mentioning these modulatory areas, it has been suggested that in migraine and probably in other headaches, that there is enhanced pain facilitation via the ascending pathways, the one I explained in previous slide, and reduced descending pain inhibition from these areas. And because of that, migraine is considered a status of hyperexcitability. So after we talked about the basic anatomical and physiological structure of migraine, let's now dissect migraine attack and look at the suggested pathophysiology of each phase of migraine. And as you know from the neurophysiological and phenotypical studies, migraine attack may go through 4 phases: premonitory or prodrome, aura, headache, and postdrome, as you see in this figure. The premonitory phase of migraine can begin as early as 3 days before a migraine headache. Common symptoms experienced during this phase, including fatigue, mood changes, food cravings, yawning, muscle tenderness, and photophobia. All these symptoms point to the involvement of the hypothalamus, brainstem, limbic system, and certain cortical areas during the early stage of an attack. As you see in these 2 figures, there are 2 processes that believed to be involved in the pathophysiology of premonitory phase. First one is the activation of the meningeal nociceptors by increased parasympathetic activity, as you see on the right figure. And the second one is through the modulation of nociceptive signals from the brainstem as well as the thalamus to the cortex, as we discussed in the previous slide. This modulation facilitate pain and reduce the pain inhibition. Now let's move to the second phase of migraine attack, which is aura. Approximately 1/3 of migraine attacks are preceded by aura, and the most prevalent aura symptoms are visual disturbances. Migraine with aura has been found to correlate with the neurophysiological wave called the cortical spreading depression. This is a propagating wave of depolarization within the neuronal and glial cell membranes. This type of depolarization is followed by inhibition of cortical activity for up to 30 minutes and coinciding with the initiation and progression of aura symptoms. In functional MRI studies, the cortical spreading depression wave have been found to be associated with a wave of hyperemia followed by a prolonged phase of cortical oligemia, as you see in this figure. In terms of the molecular basis of the cortical spreading depression, it has been suggested that the onset of this wave is caused by the extracellular elevation of potassium due to the neuronal hyperexcitability in the cerebral cortex. This would lead to the depolarization and spreading of this wave through neuronal and glial gap junction. There has been increasing evidence from animal studies that cortical spreading depression wave can activate directly and indirectly the trigeminal vascular system and trigger migraine attack, as we mentioned in previous slides. Now let's move to the third phase, which is the most important phase of migraine attack, which is headache. The characteristic throbbing pain of migraine headache is now widely accepted to be the result of the trigeminal vascular pathway activation. And this activation begins peripherally when nociceptive neurons that innervate the dura mater are stimulated and release vasoactive neuropeptides such as calcitonin gene-related peptide CGRP and pituitary adenylate cyclase activating polypeptide 38, PACAP 38, causing signaling along the trigeminal vascular pathway. In addition, some believe that cortical spreading depression initiates the activation of meningeal nociceptors through the release of molecules such as ATP, glutamate, hydrogen ion, CGRP, and nitrous oxide that are released locally during the cortical spreading depression and thought to diffuse toward the dural vessels and activate meningeal nociceptors. Moving to the final phase of migraine attack, which is postdrome. During the postdrome phase, patients experience many non-headache symptoms similar to the ones experienced during the premonitory phase, such as fatigue, difficulty with concentration, mood changes, frequent urination, and constipation, as you see in this table. More than 80% of migraine patients experience postdrome phase during migraine attack. Because many of the postdrome phases symptoms are related to homeostasis, it is suggested that hypothalamus and frontal lobe play a major role in this phase. However, widespread involvement of cortical and subcortical structures have also been demonstrated in functional imaging. Now, after we have discussed the basic anatomical and physiological substrate of migraine and migraine phases, let's discuss before we conclude another important component of migraine pathophysiology, which is peripheral and central sensitization. So in peripheral sensitization, the trigeminal vascular neurons get activated by endogenous mediators and became sensitized to dural stimuli, and their threshold for response decreases and the magnitude of their response increases, which facilitate future attacks. And this phenomenon has been demonstrated in several animal studies, and CGRP release has been implicated in the initiation and maintenance of peripheral sensitization. Regarding central sensitization, we can see it in central trigeminal vascular neurons in the trigeminal cervical complex, as well as thalamic nuclei, and they are responsible for the cephalic and extracephalic allodynia. Sensitization causes an increase in spontaneous neuronal activity and heightened response to innoxious cephalic and extracephalic stimuli, which indicate that there is persistent central sensitization and increased risk of frequent migraine attacks and the risk for developing chronic migraine. At the end, I would like to thank you for listening to this presentation. I'd like also to thank Dr. Suhaila Rukn and the organizer of the Emirates Neurology virtual conference for inviting me to contribute in this great